Thank you, Ned. That was terrific. And we're going to continue the discussion of cancer research from a national perspective with our next speaker, who is Francis Collins, joining us from Washington in his office remotely. Francis, thank you for being with us, taking the time out of what is undoubtedly a very, very busy schedule. I think Francis is known to all of you. He's been a leading voice in this country at a national level in biomedical research, science, and technology for nearly 30 years. He's currently science advisor to the president, as well as co-chair of the President's Council of Advisors in Science and Technology, PCAST. Many of you know that until recently he was the director of the National Institutes of Health, a position he held for more than 12 years, serving under 3 presidents. What some of you might not know is that his service at the national level began 15 years before that. He became the director of the National Human Genome Research Institute back in 1993, and he held that position for 15 years, and he oversaw the federal effort to map the human genome and ultimately sequence the human genome. So tremendous contributions from Francis nationally on very important issues related to what we all do in science and technology. Prior to joining the federal government, Francis was an academic. He had his academic position at the University of Michigan, and there he did groundbreaking research on mapping the human genome and using those methods to clone very important genes, disease genes, including the genes responsible for cystic fibrosis, Huntington's disease, neurofibromatosis type 1, and many, many, many others. It's a real pleasure to have Francis join us this morning to talk about cancer research perspective from the White House. And I'll just say lastly, and some of you don't know this, Francis is also a talented musician, and I'm guessing, even though it's fuzzed out there, somewhere in the background in his office is a guitar, and maybe he'll even grace us with a song. I'm not sure. Francis Collins.
What a setup, Tyler. No, I'm afraid I'm in the White House office. It's rather stiff and formal. There will be no guitar performance this morning, but I am glad to be part of this really remarkable symposium to celebrate 10 years of the Koch Institute. And Tyler, thank you for your leadership of this. I hope your retirement is more successful than mine has turned out to be. So good luck with that. So also want to thank Ned Sharpless. It's an honor, Ned, to speak right after you. It was a joy to have you as a colleague at NIH. You were a fantastic leader at NCI, and in the time you were also called upon to be the acting FDA commissioner. And we all miss you from that important role you played, and wish you all the best. And that was a great talk, and it will make mine a little easier because there are things that I was going to touch on that I think you have already covered better than I would have. We have in common, however, the fact that we are both somewhat challenged by lack of access to graphics experts. When I was the NIH director, 46,000 people worked for me, including a whole team, if I was going to give a presentation like this. In the White House, 2 people work for me, neither of whom have any graphics expertise. So this is back to the old days of coming up with your own designs. And you will see, well, they're not all that fancy, but hopefully there'll be some content there that will be worth our talking about. So I'm going to share my screen, and because I have some things that are going to decorate the conversation here. And of course, one always hopes at this point it's going to work. And I'm hoping somebody will tell me, yes, I can see your screen, and yes, it is now full screen. Are we looking good? Okay. So yeah, 10 years. And yeah, I was assigned this title, A Presidential Perspective. Oh my goodness. I guess I hadn't quite thought of myself as bringing presidential perspectives to everything I do, but I suppose that's fair, since I have been, since February, the acting science advisor to President Biden, and that is an enormous privilege. I had stepped down as NIH director in December, thinking this was going to be the kind of sabbatical year I've never had, and obviously it didn't quite turn out that way, but I'm not complaining. It is really a wonderful time to be working in this area and working for a president for whom science is absolutely the top issue that he wants to hear about and wants to be part of his legacy, his contribution. I brought some of my experience to this already in a human genome project back in the 1990s, something which I'm glad to say actually turned out pretty well. I'm seeing a little note on my screen that my internet is unstable, and I hope that is not getting in the way of my coming across. I'm just going to keep going. And we published, of course, the initial analysis of that, but I hope you saw that now, some 20 years later, after we've celebrated the completion of the human genome several times, it is now actually legitimate. Yeah, based upon the remarkable work that was done by this collaborative team, using nanopore sequencing to be able to read very long reads across those truly nasty parts of the genome, the subtelomeric areas, the centromeres, and the short arms of the acrocentric chromosomes. So we really do now have a complete telomere to telomere sequence of the human genome, which is nice to be able to see. Of course, our interest then, and your interest now, is to figure out how do we take this revolution in genomics and actually turn that into benefits for patients and provide care providers with information that can actually inform their decision-making in a way that's going to lead to a good outcome. This cartoon suggests not such a good outcome, where staring at A, C, T, and G isn't always going to be exactly giving the insights that's needed. So a lot of work had to be done to fill in all of the missing information about what those 3 billion letters were trying to tell us in both normal situations and, of course, in cancer, since cancer is a disease of the genome. So a whole lot of projects were sponsored by NIH, and these were, in most instances, international. They were designed to produce data that everybody could use, and therefore insisted that all the data be released as soon as possible after it had been cleaned up. And that really was a change in the way in which we consider the ethical responsibilities of big teams of this sort. They have to relate to access to data. Otherwise, why are we doing it? This is actually just a subset of the kinds of projects, some of them still very much going on right now, like the 4D nucleome. Up at the top, I just have to mention the Cancer Genome Atlas, because that was a very significant step forward at the time, a collaboration between the Genome Institute and the NCI that was determined to actually sequence large numbers of genomes from cancers from multiple different types and really try to understand what's the difference between a driver mutation and a passenger. And that took a whole lot of people working to come up with various parts of this that were going to be necessary for that interpretation to happen. There had to be technology development all the way through this. And there had to be a lot of analysis issue in order to really glean the facts out of this. And those of you who only recently have gotten into cancer research probably now take for granted the vast amount of data that came out of the Cancer Genome Atlas and other projects like that, including the international part of that. So a lot of that got done, and got done in a very productive way, and it built a foundation then for lots to happen. And this was maybe about the point where I thought, I've been leading the Genome Institute for a while, and I'm a little restless, and I actually left the NIH for a few months, not quite sure what I wanted to do, but that didn't last long. I got called back in August of 2009 by President Obama, asking me to consider taking on the role of directing the NIH. And that was an exhilarating experience because it was mind expanding to go beyond what I thought I knew something about, which was genomics, to pretty much everything else that relates to biomedical research. And what I found was that the NIH director has the opportunity to develop the kinds of projects that won't necessarily happen on their own without some nurturing. Projects like, for instance, the brain, to really tackle the problem of how those some 86 billion neurons between your ears are capable of doing amazing things, and what kind of technology could be invented in order to make it possible to understand that and then apply that for a long list of disorders of neurologic system. And that's been an exhilarating project, and I hope you have seen some of the outputs of that recently now with a cell census of the prefrontal cortex of both the mouse and the monkey and the human, with remarkable details emerging about how this system works. Much more to do here, but this is a profound revelation for neuroscience and for human medicine. I had a good time looking at places where we could bring together the public and the private efforts in a pre-competitive space, and that led to the founding of the Accelerating Medicines Partnership, AMP, with a whole host of projects from diabetes to Alzheimer's to rheumatoid arthritis and on to Parkinson's and schizophrenia, which have brought together the best and brightest minds around the same table to think about exactly what would make the most sense as far as a bold set of initiatives where all the data would be made available, but everybody would have a chance to work with it. And with there being skin in the game, with pharma paying half the cost. An example of that, I'll come to in a minute, is cancer and PACT. Another project I got to start, which was truly exciting to see it take shape, and now it's really taking shape, is this a program to enroll 1 million or more people living in the United States in a very diverse group and actually collect every possible kind of data with those individuals who are truly our research partners. All of us is now up to over 400,000 enrollees. Their electronic health records are available. They have had their complete genomes analyzed, about 100,000 of those now available. They keep all kinds of records of their own medical activities. They wear wearable sensors. And as this goes on, this gets more and more powerful as a means for assessing what are the factors that cause people to fall ill and what are the ones that help them stay healthy. And this is a project I think that is really coming to its own. I wanted to mention it particularly to this group because it's intended as a foundational platform for lots of other projects to be layered on top of it. I'll come back to one here in a minute. For cancer, while I was NIH director, I became totally excited, as all of you have been, about the advances in cancer immunotherapy, checkpoint inhibitors, CAR T cells, TIL cell therapy, seeing the way in which we are learning increasingly how to take the immune system to graduate school when necessary to really activate it to go after cancer that's otherwise not been recognized. And the dramatic results of that continue to appear. Let me just say that there's clearly a major challenge that lies ahead of us right now, which is how to take the successes that we've seen for things like leukemias, lymphomas, and melanoma and bring those to the solid cancers, the pancreatic cancer, the colon cancer, the breast cancer, the prostate, that still seems to be much more resistant to that approach, although people are making serious inroads. I was with Steve Rosenberg yesterday and a patient with metastatic pancreatic cancer where the TIL cell approach in the most precision medicine approach you can think of, where the cancer has been fully sequenced, the neoantigens have been identified, TIL cells have been sorted to see if there are any there that might already be activated against one of those neoantigens, expanded to billions of cells and then given back, and creating a very impressive cytokine storm, after which one hopes you will see melting away of the cancer, with some pretty dramatic success stories, but not as many as you would really hope to see. And obviously a huge problem here about how you would scale that up to the very large number of people for whom this might turn out to be the treatment of choice if it really starts to work in a very effective way. I mentioned partnerships, and with cancer immunotherapy, a partnership set up between industry and the National Cancer Institute, 11 companies involved, ongoing now, trying to make sure that we have done everything possible to identify biomarkers that might successfully predict who responds and who doesn't to a particular immunotherapy approach as a means of planning better for patient care, but also understanding the mechanisms by which this works when it works. And of course, I also had the opportunity in the Obama administration to play some role in Moonshot 1.0, which you just heard about from Ned in much more detail, and to be excited about the way this could accelerate progress in lots of directions, thanks to the Blue Ribbon panel that identified the kinds of science that seemed to be most compelling and which is ongoing, as you know. Of course, my last couple of years as NIH director were dominated, as well they should have been, with the response to COVID-19, requiring the establishment of truly unprecedented efforts to develop diagnostics, the RADx Shark Tank program, and in therapeutics for vaccines and also for treatments, the ACTIVE partnership, another public-private partnership, which I think has not gotten the amount of attention it deserves for the remarkable work that this public-private partnership did, testing out more than 20 different therapeutics in rigorous master protocol-driven clinical trials to see what worked and what didn't. Another thing that I think in my last year or so at NIH director, which I want to commend to you, is something that we should all be making top of our list as we try to assess how to make the biomedical research enterprise even more successful, is really to address the structural racism problem in the biomedical research enterprise and what we're going to do about it. And we have a couple of articles here if you're interested in reading more about the plan, something called Unite. And certainly it's wonderful to see that our major academic grantee institutions have also gotten on board with this effort, that this is not just a nice thing to have. If you're really serious about productivity, diversity has got to be involved. And we have done over time sort of serious, sincere efforts, but we have not made this the priority that it needs to be. And I think that is now our calling, all of us. Well, I stepped down as NIH director in December 2019, and then in February was asked, following the resignation of Eric Lander, to step in as the science advisor to the president, with Alondra Nelson, who was already at OSTP, taking on the role of being acting director of OSTP. Alondra and I work together extremely closely. That is a great privilege. She's a wonderful colleague in every way. This is the hallway that I walked down this morning on the way to come here in the Eisenhower Executive Office building. Yeah, that's the doorway that comes in. And yeah, this is the office I'm speaking to you right now. And I'm sorry, there is no guitar there. It's a little Spartan, although the curtains are kind of nice. And this is the place where I spend a lot of my time now running around the hallways, up and down through the floors, and occasionally over to the West Wing to try to do everything I can to take this president's vision about science, which is something he cares about deeply, and turning that into results for the American people. The assignments I was given by the president were four. First of all, there are these critical science leadership positions that need to be filled. And it sort of came about that all of these were in need of attention at about the same time. I'm not going to tell you any new news here because I'm not supposed to, but I would say that all of these are coming along pretty well. The NIH director position, there has been an intense effort to identify the most qualified next leader, and that is far enough along that I'm hoping there might actually be some news within, say, the next month. ARPA-H, which I'll come to in a minute, needs to have its director identified. There again, that search has gone quite well and is far along, and again, potentially could have some news within the next month. The person who will step into the role that is now being held in an interim basis by Alondra Nelson and me, that would be the OSTP science advisor, a cabinet position, the position that Eric Lander held. You may have seen in Science Magazine in the last 2 or 3 days, a profile of the candidate that science says is being strongly considered, and that's Arthi Prabhakar, the former leader of DARPA, and before that, the head of NIST, a remarkably capable individual, applied physics PhD from Caltech, but basically with an engineering background. But she started the biotechnology unit in DARPA. She's very diverse in her knowledge of disciplines that would be necessary for the OSTP director. So I will tell you, I'm pretty excited about this choice, and we're all waiting for some kind of official announcement, which of course is up to the president. And then there is the NCI position. Since Ned decided to move on and left on April 29th, there is an intense effort involving quite a few of us to search for the next most highly qualified candidate. And I promise you, we have some great opportunities there and people who are in fact quite interested. And I think we will come up with a wonderful new NCI director, although this one is perhaps a month or so behind the timetable of the other three, since Ned has only recently departed. So that's a little quick snapshot of how I'm spending a lot of my time. Sometimes I feel like I'm part of the White House personnel office, but it's probably okay to be sure that this effort goes forward with lots of opportunity to tap into scientific experts and make sure we do the right thing. I am also asked to serve as co-chair of PCAST, the President Council of Advisors on Science and Technology. My two co-chairs are Nobel Prize winner Frances Arnold and Vice President of Research at MIT, Maria Zuber. And let me say, they are great to work with, and we are having a pretty good time with a remarkable group of talented leaders in their own disciplines who have agreed to serve on this, about 30 people altogether. It's the biggest PCAST, which makes it a little challenging to manage, but we are, I think, making really good progress. This was the first meeting they had with the president back in January, which had to be virtual, as you can see. The next meeting with the president is coming up, we hope, according to his schedule, in July. We did have a wonderful meeting with the vice president in March. This is in the Indian Treaty Room, which is about 4 steps away from where I'm sitting right now. And of course, that's the vice president in the middle, co-chairs Francis Arnold, Maria Zuber. I'm over here in the corner. And here are all the people that are part of this remarkable group, all hidden behind their masks, so you may have trouble identifying them. The topics that we are working on right now that have been publicly disclosed and which will probably pretty soon get to the point of being specific recommendations to the president. One of them is wildfires. What could we do to have a better job of identifying and managing wildfires, which is such a huge problem, and use new technology to do that. Think about what that might look like, and you might guess where we would be going. And another is the whole problem of the shortage of semiconductors, which is turning out to be a pretty serious issue for our country, and what should be done in that space. That one is also coming along pretty well. And a long list of others that are sort of works in progress, since PCAST has only been in place for a little less than a year. And then there's ARPA-H. Ned mentioned this. This is a very high presidential priority, to create a new entity that's going to support biomedical research in a specific sort of area where you have a translational opportunity that is going to require bold and risk-taking investments, that's going to require putting together partners, many of whom might not apply to NIH in the traditional grant mechanism, and that can move swiftly, not using the traditional way in which peer review goes forward, and not be afraid to fail. So for instance, the kinds of things ARPA-H might do. Cancer vaccines comes to mind as one of those, and I see you're having a symposium about that next week, so that's timely indeed. If you want to read more about the idea here, the vision of what ARPA-H could look like, this actually was published about a year ago, but the thoughts are still quite fresh, I think, in terms of what this would look like. We've had lots and lots of consultation with lots and lots of experts, and I'm really excited about how this could play out and fill that area that currently is a little hard to do. Think what we had to do with COVID-19, for instance, with diagnostics. That was that RADx program. Boy, we learned when we had to do something in a public health emergency, we could do it really fast. That got set up 5 days after the congressional funding, and actually had identified the kind of home testing that we needed within a few months. And when you go to the pharmacy, you can see the consequences of that. But we kind of had to bend a lot of tradition in order to do that. We need the kind of framework that encourages that in lots of other situations as well. And if you go to this article, you'll see some of the ideas that people have put forward about how ARPA-H could be used, but it's going to be up to the director to decide. This is not going to be something that is manipulated by other forces. I can't imagine that cancer will not be one of the things that ARPA-H gets involved in with its $1 billion initial budget, which we hope will ramp up over the course of the coming years. And then there is also the request from the president that I assist, along with Daniel Carnival, with the reignition of the cancer moonshot, sometimes called Moonshot 2.0. And Ned has already given you a lot of information about the motivation for the way in which this has been assembled this time, and the way in which it was launched. A similar picture to what he showed you, this time with the First Lady as the speaker. And again, with the goals, the big goals here listed on the slide. But there is this 7-pillar model, which has emerged in the last few months, of what components might be part of this reignited moonshot. Diagnose cancer sooner, yes. Prevent cancer, address inequities, target the right treatments to the right patients, speed progress against the most deadly and rare cancers, including childhood cancers, supporting patients, caregivers, and survivors, and learning from all patients. Now, exactly what is going to emerge from this effort when the president rolls out his budget for fiscal year 24, which will be about next February, is still very much a work in progress. And it's a pleasure and a privilege to be part of that deliberative effort, which has involved a lot of consultation with a lot of people. Probably some of you listening have been some of those we have been hearing from. I just want to highlight a couple areas that are beginning to emerge as pretty exciting opportunities, but it is not yet decided if they will be. One of them is to diagnose cancer sooner. When you think about the way in which MCED, multi-cancer early detection, has emerged, it is both exciting and it seems desperately in need of a rigorous test to see what happens in terms of improving outcomes. Here are just three of the MCED approaches that have emerged using somewhat different technologies with different claims of how widely they will be able to detect cancer from multiple organs in asymptomatic people, all depending on cell-free DNA, although some of them are also looking at proteins, different in their way to be able to assess what tissue the cell-free DNA came from using such things as methylation. And this is just three of what may be as many as 20. We need to find out how do these work in the real world, and we need to find out if these are applied to asymptomatic people at various risk levels. What happens? Does it actually improve outcomes? We worry, of course, that it may engender a lot of investigation in people for whom the test seems to be positive, but nothing can be found. So this needs to be found out. And here's where I mentioned all of us earlier. If you wanted to think about a platform to land something like this on, here you have a group of hundreds of thousands of people who are pre-consented, who are highly interested in research and have a wide range of diversity. It seems like a pretty good idea to put them together, and that is being talked about. But this might fit rather nicely in the idea of this moonshot that is now being reignited. I'll give you one more example, and then I'll need to wind up. In the prevent cancer area, something that I will admit I've become pretty passionate about. There is a particular type of cancer that is going up, not down. I noticed that in Ned's slides. And yet it is a type of cancer which at the present time would be entirely preventable because the cause of it is actually completely curable and reversible. And what is that cancer? Well, that's liver cancer, and that's hepatitis C. And hepatitis C is currently infecting in a chronic stage about 2.4 million Americans, many of them baby boomers, the most common cause being experimentation with intravenous drugs, although many people may have also gotten this with transfusions a long time ago. Those individuals who are chronically infected are ticking time bombs. Without treatment, 20% will go on to cirrhosis, liver failure, may need a transplant, and 5% to hepatocellular carcinoma. This is the most common cause of liver cancer. And yes, it is going up. But the good news, a highly effective and safe treatment, now available from three different companies, Gilead, AbbVie, Merck, is available and FDA approved, and provides a virologic cure in 8 to 12 weeks of oral outpatient therapy with very limited side effects and a 96% success rate. How often do we see something like that happen? That's all great. We're on the way to eradication, right? Well, there's an elephant in this room. We're not. And we're not because of the realities of the initial cost of the drug, which was pegged at $80,000 to $90,000 per patient, and because a lot of the people for whom this is needed are not in the situation where they have gold standard or gold level third-party insurance. A few have, and some have gotten treated, and it's working really well. But only a small percentage of those infected with hepatitis C know they even have it, despite recommendations now that the screening should happen. And most of these are in groups with long-standing health inequities, and many don't have third-party insurance, and the prohibitive drug costs have caused many states to actually put up barriers for Medicaid patients to get access to this because they were afraid of having to pay for it. So it's time for action. And I think the time is now. A pilot project in Louisiana, which I've studied closely, focused on Medicaid patients and on prisoners, difficult areas to reach, has shown it is possible to diagnose and treat with a high level of success. The VA has done a very successful program for veterans funded by the Congress. And the drug price does not have to be ridiculously high because if one can negotiate with the company the so-called Netflix subscription model, where the company basically agrees to be paid a lump sum and then to provide drugs for anybody that you can find who is tested to be Hep C positive, then essentially the price comes down dramatically as you screen and treat more and more people, estimated down in a few $1,000s, not 80 to 90,000. In fact, that leads to an estimate with a little bit of error bar here that a national program to find and treat everybody who's positive for hepatitis C in the chronic phase would cost about $2 billion a year over 5 years. Well, yeah, that's serious money. But Harvard economist Jag Chatwal has estimated, doing careful modeling here, that this would save 110,000 people from what otherwise will be Hep C caused deaths, and half of those liver cancer, the other half from things like cirrhosis and liver failure. But notice this, when you model out what the savings is, it's about $26 billion in healthcare costs. So we have something that is not just benevolent to save lives, it actually makes total economic sense. If it's going to cost $10 billion to find and treat these folks, and then we save $26 billion in healthcare costs, the net positive here is pretty dramatic. It just seems like we got to do this. The challenge, of course, which I don't want to minimize, is that our healthcare system is not very good at paying now to avoid costs later. And that's why I'm spending a lot of my time on it. But how can we not do this? And even taking it beyond the practical, is this not a moral imperative? If we have 2.5 million people who have a disease that we know how to cure, and we're not doing everything we can to find them and treat them, what are we here for? And I'm hoping that this will find its way into the cancer moonshot, because of course, a big part of this is liver cancer, but it may be necessary to find other ways to support it as well, because it is a pretty significant price tag. I guess, as you can tell, this has become a bit of an area of intense interest to me, maybe even a little bit of an obsession, but I think an obsession that we all could embrace and maybe get together and see what we can do to save all these lives. So with that, I will draw to a close and just give you advice for the next 10 years of the Koch Institute. A few words from the author of The Little Prince. As for the future, your task is not to foresee it, but to enable it. It's really hard to foresee. But a place like the Koch can create the kind of environment to enable those discoveries that we can't even imagine now. And I wish you all the best in the next 10 years in making that happen. Thank you very much.
Francis, thank you very much for those wise and really actually inspirational words. And I'll tell you that your slides were excellent, so good job by you. Thanks again for joining us, Francis.